Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. And we have been talking about the immunology video lectures. This is a series of videos regarding different concepts of immunology. And in the last video, if you recall, we talked about the phagocytosis pathway and how different phagocytotic cells that are present in our body, they start to engulf the foreign pathogen or the part of the foreign pathogen. And one such cell is macrophage, which is the job of macrophage is to engulf the pathogen. In this case, let's say it's a bacteria. They engulf that pathogen inside. But remember, I also told you that our immune system works in two different fashion. It has two jobs to do. One is that if there is any problem with a cell, if a cell is damaged and this damage cannot be fixed, in that case, immune system will signal that cell to be killed with apoptosis or programmed cell death pathway. Now, if there is a room for improvement, if there is any chance that they can improve the situation, they can bring all those repair mechanism to fix that, but it happens less. And the second way immune system work is that if there is any pathogen found by any of the immune system cells, they will recall all the other cells of the immunity. They will just like a phone call, they will do the call for all the other cells. For example, when macrophage finds a pathogen as bacteria, for example, a bacteria take entry and macrophage engulf that. Once they engulf this, it's not the end. Macrophage's job will be end after they recall some more friends or immunity cells into this place, the friends of macrophage. Such cell examples are different T cells, different T helper cells, because those T helper cells will mediate two different sides of immunity. One is cell mediated immunity activation, another one is the humoral immunity activation. Remember, the cell mediated immunity works at the very first situations of the pathogen invasion, but it's fast, but it's not that much specific, while the antibody mediated immunity is kind of slow. It takes time to grow, but it is much more strong, it is much specific. So in this video we will be talking about antigen presentation, which is a part of that phone call that we are talking about. For example, macrophage engulf a bacteria inside. It knows this is a foreign invader, it is a pathogen, so it needs to call other cells of the immune system. So how it will do that? Now all these cells, which macrophage, dendritic cells and as well as B cells, these cells have a special capability. They not only can engulf things, they can break it down and take the portion of that fragment of the pathogen and showcase that to rest of the immune system cells with the help of a specific molecule known as major histocompatibility complex 2 or MHC-II. Okay? With the help of this is going to be like a flag, and the top of the flag they'll put the pathogen fragment and show rest of the immune system cells that this is the problem. You need to bring other cells, you need to bring T cells in there, and T cells will signal some more macrophage to come, T cell will signal B cells to come, and then the situation will be under control. So in this video we are going to talk about those cells who present the fragment of pathogen, because this fragment of pathogen is known as antigen. So as they are presenting antigen to outside to the rest of the cells, those cells are known as antigen presenting cells. Now in this video we will be talking about those antigen presenting cells, their function, and with the help of major histocompatibility complex 2 as well as with major histocompatibility complex 1, how exactly they are showcasing those fragments or antigens to the rest of the cells and what is the process. So stay tuned and watch this video. So let's talk about the process of antigen presentation and the whole pathway. Now as I told you, there are three different cells in our immune system who is acting as antigen presenting cells, and the example of these three, let me write it here, that is one macrophage, second one is the dendritic cell, and third one is the B cell. These three, they act as antigen presenting cells. Now what is the difference of this antigen presenting cell with the normal cell? Now the thing is, if I draw the difference here, let's say this is one cell, normal cell, and this is antigen presenting cell. So it's a and this is an antigen presenting cell or APC. Now what happens actually, both the cells, as a eukaryotic cell, they carry nucleus. Okay, all the cells that are present in our body with nucleus have a specific receptor with which they can take the fragments of a pathogen and show outside. So let me draw this, and that is known as MHC-I or major histocompatibility complex 1. All nucleated cells have that. As antigen presenting cells are also nucleated, so they also have that. All the nucleated cells have this MHC-I on its surface. But these APCs have something unique, some extra flag, that is a MHC-II, which is not present in rest of the other cells of our body. It's only found in those antigen presenting cells. So in these 3 cells we have one unique receptor, that is MHC-II, that will be unique there. This is MHC-II, which is not found in any other cells. Now what is the difference between these two, we'll be talking in the next video. But for now, let's understand the whole pathway of antigen presentation. The process of antigen presentation works, in this video I am going to give you an overview of how this whole process works. So there are these antigen presenting cells, macrophage, dendritic cells or B cells, take any of that. Now the idea of antigen presentation is to bring the whole immune system to the place, the whole immune cells to the place, that we found something, so bring some other cells in the place, so gather other people there. So what they do actually, let's say this is an antigen presenting cell, they have this MHC-II. For now I just erase this MHC-I because we don't need it right now to discuss. So let's only focus on MHC-II. It's have the MHC-II. Along with that, let's say this is a macrophage we are talking about. We're talking about a macrophage here. Okay, so we have this. It also have other different surface receptor molecules, for example CD80 or CD86, cluster of designation 80 and cluster of designation 86, any of that. CD80 or 86 it also have. It also have a CD40 CD40. So these are the structures a macrophage have. Now the structures they will have, the surface receptors they will have might be varying for macrophage, dendritic cells and B cells a little bit, but more or less they carry all these things, like MHC-II should be there, CD80, 86, this complex should be present there in most of the cases. Now the job of this antigen presenting cell is to interact with T cells, T helper cells, which are known as the effector T cells. You know, T cells have very important jobs to be done in the perspective of our immune system or immunity. T cells or T helper cells, they will interact with this antigen presenting cell, and the interaction between the antigen presenting cell with T cell will do 2 things, actually 3 things. There will be a crosstalk between the antigen presenting cell and T cell with the help of all these receptors. Now with the help of this interaction, T cells releases some signaling molecules that are going to proliferate this antigen presenting cells. So those signaling molecules will help to bring more antigen presenting cells there. On the other hand, those T cell will start secreting some other chemokines like interleukin 2. With the help of that, they will also bring some more T cells, and those T cell in turn can also activate B cells. So there are 3 important events after the antigen presentation. That's why this is a very important part, because this is a region where the cell mediated immunity kind of interacts with the humoral immunity, and in the junction between a cell mediated immunity and humoral immunity there is a helper T cell, and the role of helper T cell is hugely important. Okay, now let's say here there are 2 varieties of T cells found, CD4 T cells and CD8 T cells. Remember that always. 2 types of T cells are found. CD4 plus and CD8 plus. Right? CDs, and this means the different type of receptors that are found on the surface. Like there are multiple number of such receptors are found in different cell surface, and depending upon their discovery we name them CD4, CD8, CD9, like that. Now all those cells belonging to the T cells either have CD4 on the surface or CD8 on the surface. Based on the presence of CD4 and CD8 we mark them. Those who have CD4 we call them T helper cell, those who have CD8 we call them cytotoxic killer T cells. We will see their functionality in a moment. But remember here, this macrophage, let's take only one example as an APC, that is macrophage. Macrophage has MHC-II in surface, it has CD8T, CD86 on the surface, and CD40 ligand on the CD40 on the surface. Now these are the receptors that will interact with T cell receptors. So now if we draw the T cell, let's say this is the T cell, okay, this is the T cell. Now T cell have T cell receptor on its surface. We say, let's say this is a T cell receptor, also known as TCR, on the surface. It also have CD28, and it also have a CD40 ligand. So let me draw it. It has a CD40 ligand. So these three things. Now there are so many things for the interactions. You probably, if you see the books, you might find like many more interactions, and for different books different interactions. But do not confuse, this is the most basic thing that I'm going to tell you, the overview of all this process and how it's worked. So these are the three main engagement that should be present between an antigen presenting cell and a T cell for the presentation process to work properly. Now this is a T helper cell, and this is that CD4 T cell, because CD4 T cell interacts with MHC class 2. This is MHC2. CD4 interacts with MHC2, CD8 interacts with MHC1. Remember that. This is a difference. It interacts with MHC2, CD8 interacts with MHC1. So for the antigen presentation purpose, CD4 plays the vital role. So now there are three different interactions going on between an antigen presenting cell and a T cell, CD4 T cell. Now the thing is, this macrophage, it engulfed the bacteria. It starts breaking down that bacteria and break them down into fragments. Okay, so those fragments that is broken down by this macrophage is being loaded on MHC class 2. So let's say this green fragment that I'm drawing here, this is the bacterial fragment which is processed in the ER and then loaded in the MHC class 2 molecule. So now it's loaded in MHC class 2, it's supplying it to the outside, showcasing to the outside, and then CD4 T cell comes in. With the help of T cell receptor, it interacts with this MHC2 bound with antigen. That is the idea. Free MHC is not bound. MHC2 along with antigen is interacting with the T cell receptor from CD4 T cells. So once they interact, this is not making the T cell active. For making T cell activated, they need to engage two further interactions, one between CD28 of the T cell and CD8T of macrophage, and then CD40 ligand of the T cell with CD40 coming from the macrophage. While these two receptors are binding together, when these two receptors are attached with each other, that makes this bonding much stronger and much active. That is going to activate T cell. Now that is when T cell gets activated, when all these interactions are proper and attached, all these engagement are done or made. After that, when the T cell gets activated, it starts secreting chemical factors, it starts secreting different signaling molecules known as cytokines, examples interleukins. So once they start releasing all those interleukins, this T cell have two different jobs to do after this processes, actually three different jobs. it starts releasing interferon gamma or IFN gamma. This interferon gamma is going to interact and ultimately signal macrophage to proliferate. So more macrophage cells will come in this place and start engulfing the bacteria, breaking down and showcasing them. So this process is now increased, is amplified. It will also release interleukin 10, both of them. Interleukin 10 and interferon gamma are going to activate the macrophage and it will bring more macrophage, helps in the macrophage proliferation. It now also secretes interleukin 2, IL-2. When it starts secreting interleukin 2, this is going to bring some more T cells. Remember that. This is going to bring some more T cells into the location. So some gathering of T cell is also going on. And the third part of the reaction is that this T cell is secreting interleukin 4-5, that is going to activate and bring B cells. So this in a sense is the overall reaction of the antigen presentation. So you see, immune system is not limited by engulfing a bacteria or killing the cells or killing the bacteria. They also want this process to be amplified so that more immune cells comes in, say they will join, they will start functioning. Antigen presenting cell present the antigen, CD4 TCA with the help of TCR recognizes that, but with the help of this proper interaction with CD28 and 8T, CD40 ligand with CD40, the T cell gets activated, releases interferon gamma, interleukin 10. That activates macrophage, some more macrophage proliferation is there. They secrete interleukin 2, which brings more T cells. They start secreting interleukin 4 and 5, that activates the B cells, and in all sense it's contacting with the humoral mode of immunity. It brings more cells to boost up the cell mediated immunity. So the role of antigen presentation is hugely important. So this is one type of antigen presentation. Remember, there is a second type of antigen presentation with the help of another type of major histocompatibility complex, that is MHC-I, as I told you. And MHC-I is found in all nucleated cells, which I also told you earlier. So normally, let's say a cell is infected by a virus. So virus is infecting the cell and the cell is damaged. There is no way that cell can survive. Even if it survives, that virus particles can spread through that cell to the rest of the neighboring cells. So in these situations, body don't want that cell to be lived. They want that cell to be killed. For those conditions, they uses MHC-I mediated response to kill that target infected cell. How exactly? Let me show you. So just focus on here in this direction, this part of the board, because in this case, let's say this is the cell. This is the cell which is infected. Okay, this is the infected cell, and as the cell is infected, it's not feeling good and all these things are going bad. So what happens normally on the surface of every single cell, there are so many different receptors, glycoproteins that are found on the surface. Now those glycoproteins and stuff that are found on the surface of the cells, which are specific, that some of this part should be present towards the cytosolic side, some may present outside the cytosol. But this is known as the lipid asymmetry, that some part of the lipid molecule should be, or the glycoprotein should be inside, some will be outside. But this asymmetry gets altered whenever the cell had any issue like damages, or if the cell is getting tumorous, like malignant type, or if it is like HIV infects the cell or whatever. Whatever the mean is, the cell's lipid asymmetry gets altered, and as a result of that, cell gets some nearby cells, which are the killing cells, get the signal. One such killing cell is the natural killer cell. Whenever they sense something like that, they will kill the cell there. But in this case, we are telling that a virus infects the cell. So now the viral particles are present everywhere inside the cell. The cell is very sick and should be killed right away. Otherwise, these particles can spread through that cell to the nearby cells. So in these situations, you know, this all nucleated cell have MHC-I or MHC class 1. So with the help of this MHC class 1, they will showcase some part of the viral fragments, some part of these viral fragments outside. So as they showcase this viral fragments outside with the help of MHC class 1, we know MHC class 1 is detected with CD8 type of T cells, and CD8 T cells are known as cytotoxic T cells or killer T cells, because the job for cytotoxic T cell to kill the target cell. So those T cell will come now. So let me draw that. This is the T cell, and this is a cytotoxic T cell with CD8 plus. So now cytotoxic T cell will engage the interaction with the MHC-I, which is attached to the antigen, with the help of the T cell receptor. Once this interaction is properly attached, they will also need proper confirmation by interacting with other interactions, just like you see here, CD40 with CD40 ligand, CD28 with CD80. There are some other type of CDs that are present, cluster of designation molecules that are present on the surface. I'm not going to talk about that. But after binding with this MHC-I by the TCR, they also need some other accessory interactions which will be confirming this attachment. Once this attach is confirmed, then the CD8 T cells, they will start releasing some chemical factors, because upon this proper engagement and binding, the CD8 T cell undergoes a cytoskeleton rearrangement. With the help of this cytoskeleton rearrangement, all those vesicles that are present inside the T cell will be targeted to be fused with the membrane, and those vesicles are filled with so many components, mainly two different components known as perforins and granzymes. While perforins, when they release these granules, perforins will create pores in the membrane, and granzymes will take entry. Let's say these black dots are granzymes. So granzymes will take entry. It will degrade. It will activate the mitochondrial pathway of apoptosis. It will also start degrading the DNA or any nucleic acid material present there. So as a result of that, this target cell will be killed. This is a cell mediated apoptosis by cytotoxic killer T cells. We'll talk about this in much more details in the later videos. But these are the situations. So the presentation of antigen can be brought by two different ways, with the help of MHC class 2, with the help of MHC class 1. In both these cases, they have two different consequences. If we are going towards to kill a cell, then in the interaction is with CD8 T cells and MHC class 1, and if you are going to bring some more immune system to boost up the immunity in there and to call for activating the humoral mode of immunity, then we will activate through the CD4 T cells with the MHC2 molecule. So that in a sense is the antigen presentation, and these are the antigen presenting cells. Macrophage act as an antigen presenting cell, dendritic cells are acting as an antigen presenting cell, and B cells. You know, B cells also have MHC2. They have MHC-I because it's nucleated cell, and it also have a B cell receptor, which is nothing but an antibody, because these B cells start to produce antibody when it's converted into plasma cells, and when it gets converted to the plasma cells, when these T cells start releasing those, I erase that though, they start releasing interleukin 4 and 5, that will ultimately help differentiation of the B cells and then start making plasma cells. So that in a sense is the heart of this whole immune system that is conveying the message to both cell mediated immunity as well as the humoral mode of immunity. So I hope you understand the antigen presenting cells and the role of antigen presentation in immunity. If you like this video, please hit the like button, subscribe to my channel to get more videos like that, and share this video with your friends. Thank you.